All right. Hello, everybody. I hope we have a good number of people so far. So we're going to get started. I wanted to continue where we left off yesterday. And it was the part where I was showing you some of the raw data that we get from, in this case, the 10x protocol. And I believe I showed you some of the FASTQ files and what they look like, and the reads and so on. And here I wanted to start by just mentioning that this type of data, the 10x single cell RNA sequencing data, it comes with a software called Cell Ranger. And the amazing thing about this software is that all those steps that go from raw FASTQ files to align data to get feature counts, it can perform all of that in a single step. So I ran Cell Ranger onto that data, and I just wanted to show you the feature count, which will be our starting point today. So I wanted to show you what this feature count matrix looks like, which I already have it here. And you're probably expecting a matrix, with a certain number of rows, certain number of columns. But instead, you get this very long list, which is actually a list of coordinates, because what we'll see here is that in this matrix, most of the entries of the matrix are going to be empty or have a value of 0. So in a typical feature matrix data from single cell, we're going to get a lot of empty entries. So it's actually a more convenient representation to show your matrix in this format, where the first two numbers are the coordinates, so row 47 and column 1, and the third number is the value at that particular entry. We'll see this in more detail when we start with the R demo. But for now, I just wanted to give you an idea of what's the input data that we're working with. And another interesting output that we get from Cell Ranger is that it will give you a list of all the barcodes. So if you remember from yesterday, the barcode is the identifier for every cell that we're going to be working with. So here you can think of this as the names for all the cells. And I believe there are over 2,000 cells that we're going to be looking at. And lastly, we have our features. So in this case, the features are the genes. So here we have the gene ID, the gene name, and the metric, whatever we're calculating for this particular gene, which is gene expression. So I'm going to switch back to the slides. Let me change my screen. Let's go back to where I left off yesterday. Here, the demo. So like I just showed you, the matrix, the feature count matrix that we're going to work with is a matrix that is mostly empty, that has a lot of zeros, or the proper term, proper in the way that this is the name people use in the field, but at the same time not so proper because the name is a little misleading. And here we're going to review a little bit of what the dropouts are, what are the zeros in our data, and why this name may be a little misleading. So this is a figure from a review paper that was published this year, actually by a group at UCLA. And in this review paper, they look at the sources, either technical or biological, the sources of zeros in our data. So in a typical single cell RNA sequencing feature count matrix, you'll see that around 90% or even over 90% of the entries are zeros, meaning we don't have a measurement for that particular gene in that particular cell. So what they explore in this review is looking at the different steps in the library preparation, the cell isolation, sequencing, and so on, and trying to identify why some of these measurements are zero. So let's start actually from panel B. We're going to look at some of the reasons why. And as you can see in panel B, we have 6 genes over here. And we're going to explore what are the reasons why these genes may or may not have a measurement. So the first one that they indicate here is the binding of the RNA polymerase. So this is a biological zero, and this is a true biological signal, because if this is the reason why we don't get a measurement, then that means that the gene was simply not expressed in this particular cell. So this zero will be a biological zero. In our second scenario, which will be, in our second scenario, which will be the following step, we have the case where the polymerase was bound, but in the next step inside of the cell, this RNA was degraded. It could be either by some endonucleases, some enzymatic action, or there could be plenty of reasons why this could happen. But it's also a biological signal, because the gene was expressed, but it was also degraded within the cell. So we're also going to get a zero measurement for this gene, and it's also a biological zero. And as you can imagine, for gene 1, since it wasn't expressed in the first place, then of course there's nothing to degrade, so it also continues to be a zero. For gene number 3, this is the scenario where the gene was synthesized, where it survived the degradation. But then in this step of the cDNA synthesis, it was not successfully converted to DNA. So if you remember this step in our pipeline, this is where you have your RNA molecule, and then we add the reverse transcriptase enzyme. And this enzyme is going to convert your RNA into cDNA, and the cDNA is the molecule we're actually sequencing. So if your gene was expressed and not degraded, it's also possible that the conversion to cDNA was not successful. And in this case, we get a zero measurement. But however, this is a technical zero, because it has nothing to do with the biology of the cell. It has to do with our protocol, our library preparation. The fourth gene, which is the case where the gene was expressed, not degraded, it was successfully converted to cDNA, but however, it was not successfully amplified using PCR or IVT amplification. And so if you look at this diagram, you'll have your cDNA molecule, And you're also going to have some primers that are needed for PCR. And these primers will bind your DNA molecule, and once they are bound, they offer a template so the polymerase can start making copies. And one potential artifact or potential issue is that the primers never bind your DNA. So, and if you remember, the original signal is very, very small. So if the primers were not able to bind in the first place, then you're going to lose signal for this particular gene. And this is what we call a sampling zero, meaning that because of the stochasticity of the process, you're just not able to. It's not really an error from your part. It's just that given the concentration and given the amount of materials and so on, it just didn't happen that the primers were able to bind into your DNA molecule in the first place. And on our fifth gene, we have the case where it goes successfully through all of our previous steps, and it's also successfully amplified. But as you can see, here is labeled as few, meaning that even after the amplification process, there are still very few molecules for this particular gene. And so when it comes to the latter step, which is the mapping, so you've sequenced the reads and then you are aligning or mapping them to the gene of origin. Somewhere in this process, you maybe do not get enough of these reads being sequenced, or you're just not able to map them to the appropriate gene. So in that case, you're also going to end up with a sampling zero, meaning given the amount of material, in this case, the amount of reads, and the chances of a given read getting aligned, it just did not happen that this particular gene was able to get match with a number of reads. And the final scenario, which is gene number 6, is the case where all the steps are completed successfully, and you can see it has the label of many. And this is just to draw the difference with the previous step, which indicates that for this gene, we actually had many copies. And in this scenario, we are successfully able to obtain a non-zero signal. So so to summarize, the signal that we're going to be working with today is coming from the genes that were successfully able to complete all of this series of steps. And all the remainder of genes are going to have a zero signal. Is there any question about this? Okay. Yeah, somebody asks if I could share my slides for reference. Yes, all the material will be shared at the end of the course, so you don't need to worry about that. Okay, moving on. I think with this, we complete the material for day 1, actually. So now we're going to move on into the material for day 2. And to do that, I wanted to revisit, if you remember the slide I showed yesterday for the whole pipeline, all the steps in the single cell RNA sequencing data analysis. You may remember these are the last 2 panels, which is going from this expression matrix, which is the file I just showed on Huffman, and where it contains all these zeros from many genes and different cells. In that diagram, it shows how we go from this expression matrix into an actual analysis. And I just wanted to start by illustrating that this diagram, of course, is oversimplified. And what it looks like in reality is something more like this. The process of going from a feature count matrix all the way to making some meaningful clustering of cells can be divided into 6 main steps, and each step has its own things to look for, its own concepts that we're going to learn and that we're also going to apply with the code that I shared. So before we start with the first part of the data analysis, I think this is a good point to make sure we're all on the same page in terms of our RStudio setup and the data and so on. Somebody's asking what publications this slide for. Oh, I didn't include a reference. I forget at the moment because I forgot to include the reference. But I can, when I share the slides with you guys, I'll update it. So can we do a show of hands? See how many people have RStudio or another our platform already installed. Okay. I'm hoping it will be everybody. We have at least a majority. And you can lower your hands now. And we'll do a second show of hands. How many of you have a version of R greater than 4 installed? Okay. Okay, that's great. And lastly, how many of you have the version of SURAT installed? Great. Okay, so I'm gonna share my RStudio with you guys, and then we're going to. Oh, sorry. One last question. I shared a link to a Google Drive folder yesterday, which contained the code and contained the data set. So can we do one last show of hands? How many of you downloaded both of these files? Great. Great. So in the past, we also tried to work around those that had struggled to install the RStudio or the package or any of the requirements. But unfortunately, because doing so takes so much time, and it's like every person has a different issue with the platform. So we end up losing a lot of time trying to fix everybody's issues. So if for whatever reason you were not able to get any of these requirements installed or downloaded, you can still take, I'm still going to run all the steps here, so you'll still be able to see them. And I will just encourage you to take as many notes as possible so that when you do manage to install it, you'll know how to run the different steps. Let me switch my screen to my RStudio.
Branded, but you don't even have to mention single cell RNA. Yeah, no, I come up with some fancy topic or something. Okay. I'll come up with
And
maybe
let's
like
start
180
from the beginning.
target populations or something.
So here I already have, yeah, there is a question. Okay, please be mindful of your, if you raise your hand, make sure you, yeah, if you raise your hand, please make sure you put it down afterwards, because I get notifications of this person raised their hand, and a lot of times they just forgot to lower it. Yeah. There's another question saying, I only see HG19. This is the reference. YJ, where do you see HG19? What are you talking about? You can continue. You can rewrite your question in the chat, please, so we can address that. For now, we're going to start. You can open your RStudio, and the first thing will be to load the code, which is this RMD file. The way you'll do that is simply going to File, New File, or no, sorry, Open File. And here you open it from the folder where you've saved it. All right, so, okay, so the question was the HG19, that's the name of the folder. Yeah, so if you open the folder named HG19, that's a folder that actually contains the 3 files that we're going to be working with. We'll get to that. Okay, hopefully everybody has the code already loaded in their RStudio. This code was put together from the people that developed SURAT, and you can find this and many other tutorials on their website. The SURAT package is very comprehensive. It performs pretty much any analysis related to single-cell RNA sequencing data, and it's also very user-friendly and easy to execute. So we're going to be following the steps of this tutorial one by one. And another reason why I chose this particular one is that all the steps are fairly standard, meaning that whatever study that you're performing, you can imagine that from study to study, you may want to run different types of analysis, but there are some analyses that are consistent, like across the board, and those are the ones that we're going to be learning today. So once you have loaded this notebook, if you see over here, you have this green arrow. This green arrow is going to run the code for this cell. So this section over here, they're called cells, and you can just go ahead and press it, and you'll get a green bar over here that shows the progress. And this first cell should run fairly quickly. It's just loading the knitr package, which is just going to be this module is just going to be involved with the different plots that we generate and the error messages and just some other setup for our code. Okay, hopefully everybody has run this first cell. Please, you can raise your hand if you've had an issue at any point. If I don't see hands, I'll assume everybody got it running. Okay, moving on. Here we see a little bit of text, a little bit of description. Here's a little description of the data set that we're going to be working with. So the data that I share with you guys. And this data set is from peripheral blood mononuclear cells, pbmc. This is freely available at the 10x Genomics website. It's a public data set for anybody to download and play with. And this data set is made, contains 2,700 single cells. And it was sequenced using this platform, the Illumina NextSeq 500. And here is a link to the data set. So you don't need to go to this link. I'll show you here later how to access the data. Yeah, so here is a little more of description. Now we don't need to read the whole thing. So yeah, so actually let's just skip directly to the part where we read the files. First, we're going to load these three libraries, dplyr, SURAT, and patchwork. And after that, this command read 10x, this is the function that will read the input files. So all you need to do is here I wrote the path to the directory in my computer where I save the files. So you'll need to change this path to the directory where you save it on your own computer. And after you have changed this parameter, you can run the entire cell. Okay, I think we can wait a few minutes for everybody to update their path and run the cell. After, let's say after 2 minutes, we'll move on to the next step. Or you can let me know if you need more time. In the meantime, I guess for those of you that are using RStudio for the first time, some of the main features that you'll need to just be aware for this workshop would be that this layout in your software may be different from mine. So in my RStudio, I have the script panel. So the panel where the code is being written here on the left. And here on the top right, I have the console. So this is the part where we can see the output of the code that we're running. But because this is a notebook, if we generate a plot or if we print something, we're also going to see the output here, right underneath the cell. Seeing somebody raise their hand. Somebody
I have a question about how you get the path, the location path. Like how
okay. Yeah. So the easiest way you can do it, in my opinion, is that you can go to this terminal panel. If you're familiar with the Unix command line, you can, if you put CD, this will take you to your root directory, which in my case is -user-Giovanni. And you can do ls to list all the folders in that directory. So In my case, I have it in my desktop. So I do ls and in the classes software, oh, sorry, in the classes folder and the single cell RNA-seq. And yeah, so the full path will include the name of the folder with the file. So the filter gene BC matrices and then HG19. So, right, so this is a folder. So once you find the software, oh, sorry, the folder where you save the files, one way you can do it is you can move to that folder with the cd command, and then you just type pwd, which shows you the entire path up to your current location. This is one way if you have some knowledge of the Linux command line. In a Mac computer, I think if you right-click the file, it should show you. Let me double check that one. Could you, can people raise their hand if you're using a PC? Okay. Okay, we have a few people. And I assume the rest of you are using a MacBook. Can we do another? Can you please raise your hand? I see. Okay, yeah. So what I forgot to mention is that this Unix command line, what I was doing here in the terminal, this will work if you have a Unix-based computer, like a MacBook. If you have a PC, I'm not 100% certain. There was another question. YJ? Oh, okay, yeah, so somebody typed for PC, you need to replace the backslash with the other slash in directory name. So you're talking about this path. Okay, are people still struggling with the path? Can we do a show of hands? Okay, somebody has a thumbs up. I don't know if that means they managed to or they have a question. Okay, I think we should get moving on from this step. We have quite a lot of material to cover, and is there anybody who would like just one more minute to figure out this first part? Everything after this should be much easier, I believe. Okay, so we should get moving on. Just a little bit of explanation of this part of the code. So you're reading the directory where you have the 3 files that you downloaded. And this function is already expecting the 3 files and the format, and it's saving all of that information into this object, which is called pbmc.data. And if you're familiar with any R package, usually what is standard is to read some raw files and convert it into an object that the program is familiar with. And then with that object, you can perform all of the functions or processing steps that you want to carry out. So here we're actually explicitly creating this object, which is called a SURAT object. And this is the function. It's just reading the PMC data, which contains our 3 files. We're giving it a name and a minimum number of cells and features or genes that we're expecting. And here we're printing just to have a little report on what this pbmc object is. We have a total of 13,000 features, so more than 13,000 genes that were identified across 2,700 samples. So this we knew from the beginning that we're dealing with 2,700 cells within one assay. And there is the option to upload multiple experiments, multiple samples, and so on. But here we're just dealing with one sample. And our first exercise will be very simple. We're just going to print 3 rows of our data set and 30 columns. So we're in the following cell, if you run, if you press the green arrow, you're going to run this subset of the data, and you should get an output like this one. I believe there was a question. Okay. There is no question, so please run the second cell. As you can see, all of our rows have names, and the rows correspond to genes, and the columns represent cells. So what we're doing here is just printing the rows that correspond to these 3 different genes, and show me the value for the first 30 cells. And you should get a small matrix that looks like this. And I think once again, you can appreciate the fact that in this case, just for the printing purposes, just for you to view this matrix, they replace the 0 with a dot. So you can also have, actually these genes were selected intentionally because they have, it's known they are expressed in a good number of cells. If you were to select other genes, you will likely see a bigger density of zeros. Okay, hopefully so everybody completed this step. Now the following step over here, we're still doing, we're still getting familiarized with our data set. And this is just a quick, like a footnote in terms of the matrix. If you run this, the third cell, you see that these functions are just getting the size of the matrices, as if the size, as in how many bytes it takes. How big is this object for your computer? And the first object, which is a dense size, this is converting the original matrix into a standard matrix, meaning that it will show the entire data set. So if you have 100 genes and 100 cells, then it will show you all 10,000 entries. And the second object, which is the sparse size, this is the original format of the matrix, which, if you remember, it only shows you the coordinates for the entries that have a value other than 0, which should be a lot more compressed, a lot smaller. And what we're showing here is the size for these two objects and the compression ratio. And what you can see is that this sparse matrix format, what number? This is 29 million bytes compared to 700 million bytes, is roughly a 24 times compression. Like I mentioned, this is a quick side note, just to show you the advantage of the format that they chose in order to represent the matrix. Is there any question so far? Okay. So moving on, this is where we actually get started to do some analysis. And before we move to this line of code, I'm going to switch back to the slides because the first part of this code is dealing with the quality control. So we're going to review some of the metrics that we use in order to perform quality control. And once we're familiarized with the concept, we're going to come back to R and run the code. Yeah, so that will be the first part of our analysis, of our data analysis, which is quality control, over here. So quality control, this pretty much, when you're doing any type of data analysis, the very first step should be to make sure that your data is good, that your data makes sense. If you start with data that's too contaminated or too messy, then whatever conclusion you get out of it is probably not going to be very reliable. So one of the questions that we can ask about our single-cell RNA sequencing data is, how many genes do we expect? If we look at an individual cell, how many genes should be expressed within a particular cell? There are things such as too many or too few. If you have too few genes, then that could indicate that this particular cell, either the droplet was empty, there was no cell in the droplet in the first place, or it was a low quality cell, meaning this cell, it was already going some apoptosis or some other pathway that, or it already stopped transcribing, or maybe some technical reason why this cell does not contain enough RNA. So whatever measurement you get from this cell is not going to be too reliable. If it has too few genes, or if it has too many, this could be an indication of a duplet, or in some cases, multiplets. And we briefly mentioned this yesterday. And if you remember the definition, this is the case where within every droplet, you have more than one cell. And there are more sophisticated methods to get rid of duplets or multiplets based on the transcription profile. So in summary, just if the transcription profile looks like a combination of the transcription profile of two different cells, then it would assume it was a duplet. That's more sophisticated the easier way is just to get rid of the cells that have too many genes expressed. And now you may wonder, okay, but what is too few? What is too many? The short answer to that question is whatever is an outlier in our distribution. We're going to see that in more detail in our code, but for now, too many or too few will get a distribution from all the cells. And you can imagine that in average, a particular cell has, in average, all your cells have around 1,000 genes expressed. So if a particular cell has 10,000, that's clearly too much. It's clearly an outlier. So it will be very dependent on the data set that you're working with. We'll look at the actual numbers when we run the code. And at this point, I also wanted to introduce the technical terms that we're going to be using, especially with the SURAT package, such as feature count, which refers to the number of genes, and RNA count, which refers to the number of RNA molecules. And if you remember, it's also the number of unique UMIs. Another aspect of quality control is that you want to check for mitochondrial RNA. So if you remember, mitochondria is an organelle that is found within the cell, and the mitochondria has its own genome, and it has its own RNAs too. And what happens is that usually in the step where you dissociate cells from the tissue, if you remember this from yesterday, I mentioned that this is done using certain reagents, sometimes using certain enzymes that is going to break the connection between cells. And what happens usually is that sometimes these conditions are too harsh. So not only do you separate cells, but sometimes you may also break some of them. You're going to break the cell membrane. And when you do break your cell membrane, all the contents, including proteins, organelles, RNA, whatever was found in the cytoplasm, is going to get diluted in the solution. However, your mitochondria has its own cell membrane. So instead of capturing a cell, you're going to be capturing a mitochondria. And yeah, so and you can imagine that when you measure your RNA and you notice a lot of the RNA that you have for this particular cell is coming from mitochondrial genes, then that's a sign that your cell suffered during the process of dissociation. That's a sign of a lot of the original RNA was lost, and now what you're reading is a lot of mitochondrial RNA. So just to reiterate, the metrics that we're going to be looking at for quality control are RNA count. This is, we're going to be looking if we have enough RNA molecules for every cell in the first place. Feature count, this is also, this is the number of genes. So we're also going to make sure we don't have too many or too few genes expressed for a particular cell. And we're also going to look at the mitochondrial content. Like I just said, we want to get rid of the cells that have too much mitochondrial RNA. And the way we're going to approach this, of course, is by identifying and discarding the outliers. And a very important side note for this is that different samples, of course, may require different cutoffs. So if for sample one, we decide that 5,000 genes is too many, then that number 5,000 may not necessarily be a good number for sample number two. And like I mentioned, this is very data driven, and you need to look at the distribution in order to come up with those cutoffs. So let's jump back to our code. I think I'm just going to share the entire desktop. It should be easier. Right, so we're back in our studio, and we're at this point around line 70. And here's a brief description of the quality control metrics that we just went through. We don't need to do that again. So in the next cell, this is the part where we're calculating the percentage of mitochondrial RNA. So the way you can think of this line of code is that here we're selecting the genes that have this pattern MT, because I'm not sure if all, but many of the mitochondrial gene names start with MT. So we're selecting all the mitochondrial genes that follow this pattern, and we're going to calculate a percentage feature. This percentage feature is the number of UMIs, number of RNA molecules that map to these genes over the total. And maybe if somebody can help me with this part. I'm not sure if this last line of code is included in your version. Can somebody tell me?
No.
Okay, right. So you don't have this last line of code. We're going to plot the values later anyway, but if you wish, you can add this line afterwards. I'm going to paste it in the chat. Where is the chat? Right. So when you run this cell, it's going to calculate the percentage of mitochondrial RNAs. And then this new line of code I just shared with you guys is plotting a histogram. So we have a distribution of this percentage of mitochondrial genes, mitochondrial RNAs. And as you can see from the plot that it generates, we see that most of the counts are less than 5%, meaning most of our cells have less than 5% mitochondrial RNA, which, you know, is something good. Usually 5% is kind of like the arbitrary cutoff for what we consider too much mitochondrial RNA. But like I said, it's kind of arbitrary. Some people may decide to be more stringent or more permissive with this number. Okay, so hopefully everybody's done running this cell. We can move on to the next one. And the next one is just going to print our data table. It's going to print the first 5 rows. So this is just to show you what are the information, what are the values that we're using here. And if you print, you see that the different rows are different barcodes, different cells, and they're all coming from the same study. We have our RNA count, so number of unique RNA molecules. We have our feature count, which is the number of genes that are being expressed. And then we have our mitochondrial percentage, which based on this distribution should be somewhere between, mostly between 0 to 5%. Oh, somebody has a question. Brian? You don't see, are you talking about the histogram? Oh yeah, I'm sorry. I sent the extra line of code to one student only. I'm sorry. Okay, this should get to everybody. Yeah, thanks, Brian. Yes, and
I just wanted to know what's the difference between count RNA and feature RNA?
yeah, so if you remember, feature is actually genes. So feature RNA is actually the number of genes that are expressed. And count RNA is the number of RNA molecules. So you can see that the count RNA number should always be greater than feature RNA. Okay, hopefully everybody has completed with this cell and has taken a look at our data frame. And let's complete this last part, and then after this we're going to take a little break. So, oh, there's another question. Count feature means cell number. Count. So feature means gene. Yeah, so before I think I mentioned feature count as the number of genes, but here actually it's labeled as feature RNA. I think at some other points it's gonna be labeled as feature count. But feature count or feature RNA is referring to the number of genes. Just to clarify, okay. And we can go ahead and run the next cell around line 101. Also, if this is going to generate, I believe one plot. Yes, one plot, two plots. And you can click on them to view them. And if it looks too funny, like too narrow or too wide or whatever, you can change these parameters up here, figure height and figure width to 4 and 7. That's what I'm using over here. So you get a nicer plot. So once you have run the cell, you should get two plots. And we'll look at the first one, where this first plot is actually divided into 3 panels, where you have your feature RNA, your count RNA, and your percentage mitochondria. And from this distribution, if you haven't seen a violin plot before, in this case, the violin is behind all of these dots, but hopefully you can see this pink shape that represents the distribution of the dots. So most of them will be around this value, and we'll get fewer as we go higher up. So as you can see from this distribution is that in terms of feature count or gene count, we see that most of the cells actually have somewhere between not 0, but close to 0, all the way up to 2000, roughly captures most of the cells. And we have a few outliers here that have more than 2000 genes. If we look at the count RNA, similarly, we see that most of the cells are in this range, which is around 2000, maybe 2500. And then as we move higher up, greater than 5000, for example, or 7500, we'll see a few outlier cells. So yeah, we can say most of our cells have somewhere between 0 to 5000 RNAs. And this we saw before in our histogram, but this is a different representation, which is the distribution of mitochondrial RNA. And here we can see that for the vast majority, this number will be somewhere between 0 to 5%. Oh, somebody's asking if the plot function is from SURAT or ggplot. So this function is from SURAT, but I think it's using ggplot as a dependency. So in the email I sent yesterday, I asked you to install ggplot. You can still do that during the 10-minute break. I believe it shouldn't take too long to install. Yeah. So this is our first plot where right now we can make a very educated guess of what would be our cutoffs in order to get rid of outliers. And in our second plot, here we have a little more things going on. This is a scatter. The first one, we have our percentage mitochondrial RNA in the y-axis. And in the x-axis, we have our RNA count. So by looking at this plot, usually the first thing that we notice is that we see very high mitochondrial content when the RNA count is small. And which means that you can imagine from cell 1 to cell 2, both of them probably have the same amount of RNA molecules coming from RNA. RNA molecules coming from mitochondria, but one of these two cells have more RNAs overall. So, which is driving the proportion a lot higher for one of them. And of course, that's going to be a cell that you don't want to include into your study. So with a mitochondrial cutoff of 5%, you can see that we'll be getting rid of these cells over here, and which are mostly cells with a low mRNA count. And in the second panel, we have in the y-axis the feature count or the count of genes, and in the x-axis we have the RNA count. And here this plot is to show how these two variables relate to each other, and you see that it's roughly a linear relation. And if you look at the values, here the top value is around 3000, and here the top value is around 15,000. So what you can tell from this plot is that there is this tendency of having an average gene be covered by roughly 5 reads. And this plot is just to show you pretty much how this distribution of RNAs, how the RNAs are getting distributed across your genes. And yeah, in this regard, we'll probably be getting rid of the cells that have too many genes. So whatever is above, if we decide 2000 or 2500 above this line, and In this particular tutorial, we're not going to use RNA count as a matrix, but in other studies, you can, in other data sets, you can also see if maybe you have particular cells with too few or too many RNAs, then you can also set up some threshold to get rid of them. Okay, is there any question? I'll get back to you, Brian. And for the rest, we can take a 10 minutes break now. We'll continue at 2:45. Okay, so after we've performed these two plots, and we've looked at our metrics and what the distribution looked like. If you see the last line of code in this cell, so around line 108, you see that we're doing a subsetting. So we're selecting the cells that have more than 200 genes, less than 2,500. So we're drawing the line somewhere here. And that has mitochondrial RNA concentration less than 5%. 5%, so we're, in this plot, we're drawing the line over here. So as you can imagine, we're getting rid of a few cells by performing this subsetting. And then the next part of our analysis would be to normalize the data. For this, we're going to jump back to our slides, and we're going to learn some theory about data normalization. So data normalization in general means to, you want to transform the data, you want to transform the values with the purpose of make it comparable across two different samples, or in our case, across two different cells. And the main assumption that we're going to make for our normalization is that all cells have the same amount of mRNAs, of RNA molecules. Now, if you remember our plots, you know that that's not true. Some cells have more RNA, some cells have less. But we need to make this assumption to transform our data, because you can think of the scenario where for gene A, you have 10 RNA molecules in cell 1 and 10 RNA molecules in cell 2. And even though the count is the same, but those 10 molecules may represent a very different fraction, right? You can think maybe for cell 1, that's half of the transcriptome, while for cell 2, it's only a very small proportion, just another gene. So the way we normalize is by getting what proportion of the total count of RNAs is a specific gene expressed. And the way we do this is by converting our count data to this new unit that's called counts per million. And the way we convert this data is pretty straightforward. It's given by this formula. So the counts per million for gene I and cell J is going to be given by the RNA count that, that is assigned to the gene I in cell J, divided by the total RNA count or UMI count present in cell J. So here we're obtaining our proportion, and then we're multiplying this by some scale factor. The scale factor is really just to bring the data to a scale that we're comfortable with working. For the CPM, where the scale factor would be 1 million, but you can also use a different number. And because, like I mentioned, the motivation for normalization is that we want to compare values across cells. And in order to do this, we want to assume, we're assuming different cells have the same number of RNA molecules. You can think that another approach, another technique to do that would be to downsample UMIs or RNA molecules until to the point where the cells have the same count. This is a little less conventional, and I've definitely seen people do this, but there is also the issue that if you're removing UMIs, you're losing power, right? You're losing the amount of data that usually you want to preserve. It is a valid technique though. So I would just point out that that's the drawback of this approach. And then furthermore, the data gets transformed into the log scale. And the reason for this is that expression values usually span multiple orders of magnitude. So you can have certain genes that have 0 to 10 counts, very few, very lowly expressed. And then you can also have other genes that have hundreds or even, well, thousands is a little bit of a stretch, but like hundreds of reads mapping to that particular gene. And so because you have this wide range of values in gene expression, you want to transform them into the log scale so that the bigger values don't completely dominate over the smaller ones. And also whatever variance or spread there is on your distribution, you also want to convert it into the log so that it's more comparable with the more lowly expressed genes. And another side note that I wanted to make with normalization is that this is an ongoing question and in the field in single-cell RNA sequencing, because you can think that with all the traditional approaches, there is always drawbacks and there is always limitations, and these limitations, potentially affecting your final results about the cells, about whatever conclusions you're making about the cells. And nowadays, there are very sophisticated approaches for normalization, including machine learning. And I would say like every now and then there is a new paper like presenting their new approach. Like we figured out, we got it. We have this new method that will finally normalize your data for good. I'll just say that it's still a very active area of research involving a lot of bioinformatics and statistics, trying to figure out what's fair, what's the proper way to normalize in order to make fair comparisons. Yes, there is 2 questions.
Yeah, I have a question. Regarding the normalization, what we normally do for the RT-PCR, we normalize it against the housekeeping genes. Was wondering whether this is something that we can borrow from the RT-PCR and do the same here, or normally they don't use it. So
Right. Yeah. That's a good point. And actually, there are methods that use that principle. So that the idea is that there is this group of genes that you expect to be somewhat to have somewhat consistent expression across cells. So you can use them as a reference in order to have a better idea of what's considered normal and what's considered outside of your distribution. So using that reference for your normalization is actually a technique that is being used by certain methods.
I was just drawing it. I see. Yeah, thank you. So it's
and the other question, so the question is, what is the option for duplicates and triplicate samples versus control normalization? Normalizations across samples. So YJ, are you asking if, say, if I obtain a tissue from patient 1, how to normalize in order to compare it to cells from a tissue from sample from person 2 or something like that? Yeah, that's a good question. And that actually falls into data integration. And we're going to, that's actually planned for the third day of this workshop. So tomorrow we're going to be touching on that. Yeah, those are great questions. Thanks. So moving on with normalization. Yeah, so we obtain usually the log +1 because, you know, if this value is 0, the log of 0 is not defined. So we just add a 1 as a pseudo count to all the values. And in this paper, they included a very interesting illustration on the effects of normalization onto the data. And I just wanted to share how this normalization is transforming the distribution. So you can see in the leftmost panel, panel A, we have our expression units in terms of counts. This is what we obtain in our raw data in our original matrix. And if you look at the distribution in this histogram, you can see that it's very continuous. It looks like a fairly natural distribution. And this is typically what you get. Most of the genes having a 0 count or a very low count, and then trailing off when approaching higher values. However, when you perform counts per million normalization, all of a sudden see that the shape of your distribution has changed dramatically. When you normalize these values, you see that when the histogram bar at 0 it stands out from the rest of the distribution, right? And the reason for this is that all the rest of the values that are greater than 0, they are getting transformed and they're getting shifted, while 0 remains 0. And you can think of what's the problem with this data transformation is that now the difference going from 0 to higher values is no longer continuous. It becomes almost a binary problem. It's either 0 or it's something greater than 0. And the question of how the zeros, meaning what fraction of your entries are zeros, what percentage of zeros is acceptable, and like how the zeros will further influence downstream analysis, it's also an ongoing question. It's pretty known, of course, that we'll see later that the greater the amount of zeros, the less you can trust certain metrics. But for now, the point I wanted to make is that when you transform this data, you're really creating this division between the 0 counts and whatever is greater than greater than 0. And even more so when you get your log 2 of the CPM value +1. Okay, yes, so before moving to the next section of the data analysis, we jump back to our code and we will normalize our data. Like I mentioned before, the good thing about these packages is that everything is already implemented and sometimes it's very easy. So normalization can be done by running one function that is called normalized data, and it's very straightforward. So in this cell, in the top cell, we're specifying the method and our scale factor. If you remember for CPM, our scale factor is 1 million. The default for SURAT is 10,000, but we can always change this value to 1 million. And the point that they're making in this tutorial is that you don't need to always specify the parameters. In this case, these are default. So it'll be the same if you run the function with only with your SURAT object with the pbmc variable. So we can go ahead and run these two cells. And this should convert, this should convert our row counts into the CPM. Well, in this case, the M stands for million, but in this case, it's 10,000. But our CPT, well, let's still call them CPM values. That will be stored in the pbmc object. And that's it for normalization. Now we can talk about the next step in our pipeline, which is identifying highly variable features. In our case, identifying highly variable genes. So the motivation for this step, why do we want to identify the highly variable genes, is that regarding the point that was made earlier, actually, like I mentioned, there is a bunch of genes, housekeeping genes, that you expect to have a somewhat consistent expression across different cells. So you can imagine that the signal for these genes is going to be lowly variable, meaning it's going to be similar across different cells. However, we actually want to focus our attention into the genes that are changing across different cells. And by doing so, we're also making the signal stronger, because when you're analyzing the entire set of genes, the difference from cell to cell, it may be hindered by all these housekeeping genes that have similar values. And if you only focus on the highly variable genes, then you can truly appreciate what's different between the two of them. So one approach in order to identify these highly variable genes is by calculating this coefficient of variance. The way it works is that you'll generate a plot where your x-axis is the average normalized read count, in this case, our CPM expression value. And on the y-axis, you'll have the CV square value, or some expression of normalized variance. And the point of this plot is to show that when a gene is more highly expressed, the variance for that gene is smaller. And when the gene is very lowly expressed, it will tend to have a very high variance. So what do we do with this information? The next step is usually to model. We want to find this relationship between the variance and the average expression value. And the reason why we want to model, we want to find this function that describes the data, is that based on this function, we can identify what stands out. So for this particular figure, they used a gamma distribution to model this curve. But there's other methods also to use, like the moving average approximation, and there is some other statistical approaches in order to find this curve. But like I mentioned before, the goal is that we have a relationship between these two variables, so that for a given expression value, how much variance do I expect? And if a gene has more variance than that, then we'll consider it a highly variable gene. And yeah, so one question is, what is the difference between the solid line and the dashed line? The solid line is representing the trend. So this is a function that's modeling all of these data points. And the dashed line, if I remember correctly from the paper, is the confidence interval. So adding some standard error area to this curve. So whatever is right of the dashed line is what we consider very highly variable. And going to more details for this particular plot, you'll also see that we have some blue dots over here. And for this particular experiment, these blue dots are the spike-ins. This would be actually a good exercise. Maybe somebody could remind the rest of us what is a spike-in? Anybody wants to tell us the definition of a spike-in? Okay, so a spike-in, we mentioned it yesterday, is actually an RNA molecule or a set of RNAs that you introduce into your experiment. So the purpose is that they serve as controls because you already know how many molecules you're inserting. And when you complete your analysis, the sequencing and quantification and all of that, you can check the value compared to what you were expecting. And that way you can calibrate, you can test your method for accuracy. So here in this plot, the blue dots represent the spike-ins that were used in this particular experiment. You can see that the x-axis for the dots represent their original concentration, and the y-axis represents the variance observed in this experiment. So in this, the conclusion that they're making here is that, you know, this curve is modeling pretty well most of the spike-ins. And then all the dots that are more to the left over here, meaning this area over here is what is representing pretty much all the data points that are not fitting too well with our distribution. The conclusion is that this will be more likely due to some technical error. Yeah. So when it comes to identifying highly variable genes, there are some recommendations to follow. Usually people tend to select in the range of 2,000 to 5,000 genes. And if you remember, we're doing a feature selection, meaning that from this point onwards, we're not working with our entire set of genes, which was around 13,000 or something. We're only going to be working with however many we select, preferably in this range. But it's also worth noting that this is an arbitrary cutoff, meaning that there is no good reason to select these numbers exactly. And usually the best practice, and something that I really want to highlight here, is that it's good to try different values. You start selecting, say, 2,000 genes, and then you proceed with the rest of your analysis and you obtain some results. And then you want to repeat with a different number of genes. Maybe you move to 3,000 or 5,000. And then you check for consistency or how your results are different every time. And the reason why this needs to be done is, like I said, there is no robust reasoning behind how many genes are good to select to move on. And it could also vary from data set to data set. Yes, so before moving to data scaling, we're gonna jump back to our RStudio. Now we are at this section, line 120. And here we're performing our selection of of highly variable features. And just going a little bit over this code, the way to find the most variable genes using the approach that we just went through is through this function called find variable features. And the selection method VST, this is actually precisely that regression that I mentioned, where you try to correlate the mean expression value with the coefficient of variance. And number of features, this means that we're going to select the top 2,000 genes that are more variable. And then in the next line, top 10, this is just the list of the top 10 genes that are more variable. And we're going to use this list to label these genes in the plot. So we can go ahead and run this cell. And actually, this I noticed is they're both the same plot. The only difference is just that this one is labeled and this one is not. So for clarity, I'm just going to plot the second one alone. Oh, I don't know if I like this, it's too white. Anyways, let me just go back to the original plot because this is too stretched horizontally. It's not very clear. Okay, so we're going to be focusing on this plot. Notice here that it's the same plot I was mentioning before, except for the y-axis, instead of plotting the coefficient of variance, is plotting a standardized variance, meaning that this standardized variance is already the expected variance given an average gene expression. So whatever is too far from this middle value of 1 would be like highly variable. So it's pretty much the same idea. Whatever genes that are way outside of our main trend line, those are the highly variable genes. And in this case, we're labeling them with red dots, and we have 2,000. The non-variable count will be around 11,000. So together we have our original 13,000 genes. And here we're including the name of the genes that are highly variable. Here we're labeling only 10, just because we cannot fit 2,000 labels in this plot. But from this point onward, we're selecting the features of interest, and our features of interest are these 2,000 highly variable genes. Yes, so somebody saying that the graphs appear very smooshed. So you can change these parameters up here, where you specify the height and the width of your plot. We'll be moving on to scaling the data. And yeah, for that, we'll jump back to the slides. So you can think of normalization, the first type of data transformation that we perform, and then scaling, the second type. And the reason for scaling is, once again, to make values more comparable to each other, but also to the final product of scaling is that you make every gene weight the same as any other gene. What does that mean? So if you think of gene 1, for example, that tends to be very highly expressed throughout cells. And gene 1 always has the highest count, say it has 1,000 reads, whereas the other genes have very few, like less than 10. So when you perform our downstream analysis, you see that gene 1, because the values are so high, is going to strongly influence our methods of clustering and visualization and so on later on. But gene 1 may not be particularly important, to our analysis, right? We want to consider all genes equally. So one way to address this problem is to scale the data. When we scale the data, we're actually obtaining, in the first step, we're actually we're normalizing all the values divided by some total. So you can see that cell 1, for example, it has a total of 100 units or 100 row counts or whatever this may be. So you proceed to divide every value by 100. And you do the same for cell 2 and cell 3. And this way you have comparable values across for a given gene across the different cells. If you remember again, this is following the similar logic as the counts per million, the assumption that they should have the same amount of RNA, and then the expression for a given gene is just the proportion out of that total. Another aspect of scaling, which is not detailed here, and it's probably a little harder to visualize, is that you also want to have the same variance across genes. And what that means is that the variance refers to the spread of value, how different are the values across different cells. So for example, gene 1 has the same value in the 3 cells, so the variance is 0. And let's see, and gene number 2, it has 0.1, 0.1, and 0. So gene 2 is going to have a bigger variance, probably 0.06 or something like that. And so you can see that the variance for gene 2 is already different from variance for gene 1. So when you perform scaling, you're also trying to make sure the variance is uniform across the different genes. So with that, we go back to our code in R. And the way we scale the data, again, very straightforward, is just a simple function called scale data. And in this tutorial, however, they want to point out that in the first cell, they're scaling the values for all the genes, which is something you can do, but like I mentioned before, we perform feature selection with our highly variable genes. So those are the ones we care from now on. So we can run them both, it won't affect. But what we want to do at the end of the day is to scale the data using only our features of interest, which are the genes that are changing across cells. Are there any questions so far? Okay, and one interesting thing about the scaling too is that you can regress out variables that you're not interested in. Now, what do I mean by regress out? Is actually a technique in statistics, and just to very quickly summarize it, it consists on finding how much of the signal that you're seeing is due to a specific variable. So in this case, we want to see, even though we already got rid of the cells that had very high percentage of mitochondrial RNA, maybe some of the cells that remain, this mitochondrial RNA, is still affecting the overall scaling. So one very cool feature from this scale data function is that we can precisely identify how much is this percentage RNA influencing the rest of the matrix, and we can ignore it or skip it, just so it don't affect the rest of our data points. That was a very brief explanation, but there is actually a lot of statistics involved into doing this. And yeah, we won't really have time to expand too much. And then the next step would be running our PCA analysis, which is principal component. Now for this is a very, this is a key component of our analysis. So I think it would be good to take a 10 minutes break here, and then we'll start with PCA when we're back. Okay, hopefully everybody's back by now. Is there any question before we jump into PCA? Okay, so let's go back to our slides. Now we're introducing the fourth step of our analysis, which is dimensionality reduction, or PCA. PCA stands for principal component analysis, and the goal of this step is to reduce the number of dimensions of our data. Now, our data is multi-dimensional, and what we mean by this is that if we consider what values are associated for every cell, it's actually a vector, an array of values where one number or one value is the expression of a particular gene. So in that sense, we can say that our data for every cell is multidimensional. It has as many dimensions as we have genes. So the problem with multidimensional data is that, of course, it's not practical, feasible to do the proper analysis, especially because later on we're going to be clustering cells in higher dimensions. You know, things tend to be closer together, and it's really hard to figure out what's the proper way to split them. So there is this necessity to reduce the number of dimensions in order to continue with our data analysis. And so how would we go about this? Because we have every value in our array, in our vector of values, is a significant one. You know, we can just skip them. So how do we go about this? How do we reduce the number of genes? So the approach that we're going to use is to find out a metagene. This metagene will be a new feature whose value will represent as many of these genes as possible, as much as possible. So right now it sounds a little abstract. So I'll go into a little bit of the details of how PCA achieves this. And we can start with a very simple example where we have data in 2 dimensions only. And here the X and Y axis indicate your 2 dimensions. And here you have a series of data points, that have coordinates in the 2 axes. So the goal of PCA will be to find a new axis in which we can represent this data. So if we look at these projections, what are the values in the y-axis and what are the values in the x-axis? Let me change to a laser. Yeah. And we show these projections over here. We see that it's somewhat similar, right? Like the spread of points is actually similar across the 2 axes. So what PCA will do is actually try to we'll try to move this axis in a way that we can represent these points in a new coordinate system where hopefully fewer dimensions will be necessary. So in this case, we're turning our axis into a new set of coordinates. Instead of X and Y, we label them as PC1 and PC2. And if you notice what's different between these 2 different sets of coordinates is that in the PC coordinate or in the PC space, you see that across the PC1 axis, your data points are very spread out. And that's something you can see over here too. Your dots are very spread in this direction. What about the PC2 direction? So when you're looking at this axis, you see that your points are actually not as spread as in the PC1. They're actually pretty clustered within a much smaller region. And that's something you can see here in the PC2 projection. So here, what we have accomplished is that actually, if we use PC1 only, we're pretty much capturing most of the original information, right? Because the projection across the PC1 looks very similar to our original data. And PC1 is just 1 dimension as opposed to 2. And so in this way, we accomplished to reduce the number of dimensions. Now you may be wondering, what about PC2? There is still some spread, there is still some variance. Yes, that is true. If we use the combination of PC1 and PC2, then there is no dimensionality reduction, and our data is pretty much, for all intents and purposes, identical. So what I'm trying to say with this is that when we perform a PCA analysis and we select fewer dimensions than the original data, there is always going to be a little bit of a loss, but hopefully you will be able to capture much of the original patterns as possible. And to further illustrate this, I found this cool animation online, which is pretty much showing what I just explained, where you have your dots in your original S1 and S2 coordinates, and then these gold color arrows are the new coordinate systems that were calculating for this data set. And similarly, as the example before, you see that PC1 captures it captures most of the trends of the points, most of the relative position. So, and PC2 captures a lot less. So if we were only to consider PC1, then we'll still have a good percentage of the original variance. The technical definition for PCA is to find the axis that maximizes the variance of your original data set. And however, I already mentioned, one limitation of PCA, that is that you may not, if you're reducing the number of directions, losing some data losing some data, some of the original pattern is inevitable. But there is also another issue that is a little harder to visualize, but it has to do with the topology of your original data space. And so what does that mean? You can consider, for example, that your data set is in these 3-dimensional coordinates. So you have a height, it has a width, it has a length. And the pattern, the structure of your data is shaped like an S. Hopefully you can all see that this S shape is 3-dimensional because it has a width, and the data points are colored based on their relative position on this S shape. So what happens when we try to reduce this 3-dimensional topology, this 3-dimensional structure into 2 dimensions? It will most likely end up looking something like this. So you see your dark blue dots all the way at the top and bottom of the plot. In the middle of the S, we have this dark red, which are now in the middle of the plot. And it kind of looks like it's doing its job, right? You can consider one thing, though, that is not quite represented here. Because this is an S shape, if you look at the distance between these dark blue dots and the middle dark red dots, you see they're actually not as far, or at least not further than the red dots and the yellow dots, right? However, in this plot, the dark red dots are as far as possible as from the dark blue dots. So when we try to represent this topology into a lower dimensional space, you see we're losing some of this connectivity, some of this vicinity of the original data points. And this is just something inevitable when it comes to PCA, is that if the original topology of your data is too complicated, if in the high dimensional space, some dots or some data points were close together, it is very possible that after PCA, they will be further apart, and therefore losing some biological insights. So how do we go about doing that? Well, actually, I'll mention some of the strategies that we'll use for PCA in order to maintain as much of the original information as possible. But another issue that I also want to bring up, another limitation I also want to bring up with PCA, and I kind of touched at it a little bit earlier, is that when you try to, in this study, what they try to do is they obtain some spiking RNAs, and they sequence, they obtain some expression, they run some PCA, and what they show here is that how these different samples, what pattern they follow when you plot the new coordinates in the PCA1 compared to the amount of zeros that it had after sequencing. So if you remember, PCA1, is our new coordinate system. It's a combination of the expression of multiple genes in this case. And the zero fraction is just the amount of the fraction of cells that had a zero in it. And what you can see is that there is a pretty strong positive correlation. So what does that mean, though? What does it mean that the fraction of zeros correlate with your first PCA? So the first thing I should mention is that the PCs, or the dimensions, they are sorted based on significance, meaning that PC1 is going to be the coordinate that captures most of the spread of the data. PC2 captures a little less, PC3 even less, and so on. So what does it mean that the PC1 is strongly correlated to the fraction of zeros in the data? This suggests that the signal that the source of variance in our data is mostly given by how many zeros we have originally. So the first dimension in the PC space is actually capturing how many zeros are present in our data set. And you can think of this that this is potentially an issue also because you want this new coordinate space to capture patterns in gene expression. And maybe the fraction of zeros, you can think of it also as a pattern, but like we mentioned before, there is many reasons why you could have a zero. Not all of those reasons are biological. Some of those are sampling errors, some of those are technical errors. So this is potentially another issue with the PCA analysis. However, we can see that once we run our PCA on our data point, we'll be able to nicely plot it into a two-dimensional space. So originally we had something around 13,000 genes, I believe. So originally we'll need a plot with 13,000 axes in order to visualize our cells. But thanks to PCA, we can use PC1 and PC2 in order to get a two-dimensional plot. And in this particular case, what is showing you here, a very important measure, which is how much variance is captured. So PC1 is capturing around 36% and PC2, 16% of the variance. The way you can think of this is that between PC1 and PC2, you're capturing 36 plus 16, so 52% of the original spread of the original distribution of your data. And the point of this slice of this particular figure is also to show that when we ran our feature selection, when we selected our most highly variable genes, we can actually improve the percentage of variance. So sorry, it's actually going the opposite way. When we start with more genes, we have fewer variance explained. So 5 plus 13, only 18%. But when we select a smaller subset of genes, we're actually going to have a much greater percentage of the original variance into these two PCs. And lastly, when it comes to PCA, the question is, okay, so we can use two PCs to represent our data into two dimensions, but how many PCs should I actually select? Because we just saw that there are so many limitations with PCs, and these limitations are more significant the fewer PCs you select. So ideally, you want to select few PCs, so few dimensions, so that it's easy to work with, but not too few to the point that you introduce all these biases and limitations into your analysis. So we're going to look at two methods in order to select the best number of principal components. And another side note here is that similarly as other steps explained before, the selection of PCs, the number of PCs that you're selecting is also going to be something you want to carefully evaluate, and you may want to try different values as you move forward. So let's go back to our RStudio and let's see where we left off here. So after we scale the data, we move to our cell in line 165. And yeah, so the function to run PCA is called run PCA. Very straightforward. You press a green arrow. And as you can see, we keep redefining this pbmc object. And what's happening is just simply that it's adding new attributes to this. So now PBC is going to have within it some object called PCA. And you'll see that it's generating quite a lot of output. We won't go over this. I think it will become more clear later on. So after you've run that cell, we can go and run the next one, which will generate a PCA plot. And if you look at the parameters for the first part, the first function is actually going to print 5 PCs, so 5 dimensions, and it's going to print 5 genes for each PCs and how they correlate to this new axis or this new meta gene that we just came up with. So let's see what that looks like. So this is our first output. So here with this parameter, we're printing the first 5 dimensions. So we have PC1 all the way to PC5. And we're also printing 5 features, so 5 genes for each PC. And they're further split into positive and negative. So if you remember, the definition of PC is this new access system, but it's also a new meta gene. So our original genes are these ones, right? And our new meta gene will be the PCs. So what this is telling you is pretty much how the values in these original genes correlate with a new PC. So those genes that have a positive value in the new PC are these ones, and those that have a negative value on the new PC are over here. And this becomes a little more clear with the first plot. It's a little smooshed. Let me change the height for this. 5. It's a little better. Yeah. So what this plot is showing is here we have our a list of selected genes, and here we're showing what are the new values for these genes. Before they used to have a value in terms of normalized scale expression, but now they have a value in terms of PC. And for PC1, they have these values, some are positive, some are negative. For PC2, they have these other values. I don't think it's too relevant, the fact that they're positive or negative, because it's not easy to interpret. So we understand this is a new coordinate system. It also represents a meta gene, which is a combination of the values of other genes. But, you know, it's not very intuitive to really analyze, like, what does it mean to have a positive PC or negative? And it's not an information we'll use later on. And then lastly, the third output, which is this dim plot that we're making. This is plotting all of our cells. So all 2,700 minus whatever we discarded in quality control. They should be plotted into this 2-dimensional system. And this is not quite clustering yet. It's already clustering in theory, but we actually won't rely uniquely on PCA for clustering of cells. But at this point, this plot is showing you that based on the expression patterns, you can already see cells getting closer together in this PC space. And if we move on to the next cell, where we have this dim heat map function, what we're going to do is select 500 cells, and we're going to plot their values on one of the PCs, so in this case, PC1. If you plot this function, you'll see we have here our gene names, and here we're selecting, and here the values for that correspond to the different colors are the values in the PC. So I don't have the legend. I believe yellow could be like very, very high PC value and then purple very low, or the other way around. But what this is showing you over here is that now we can very clearly cluster different genes based on this new coordinate system, and that the clustering, just from looking at it, it looks like it's a very clear-cut clustering. Like these many cells have very similar expression pattern, and these cells have a different expression pattern. And further on, we can generate this plot, not just for the first PC, but we can generate it for the top 15 PCs. So we're going to see how our data projects in 15 dimensions. And once you generate this plot, I want you to notice a trend that actually becomes very evident. We've seen this plot before with the PC1, where I mentioned that the cluster looks very clear-cut. There is very strong signal for a unique pattern among this set of genes. And we can see here, this is clearly one cluster, this is clearly another one. If we look at PC2, we see that, okay, it's still pretty strong. You know, we can still tell how what the two different groups are. And then as we keep moving into these other dimensions, PC3, PC4, PC5, at some point you see that maybe around PC6, PC7, it's not so clear anymore, right? And if we go all the way down to PC15, you see it's a pretty big mess at this point. Like we can't really tell anything from this plot. So if you remember what's the value of what's the definition of every PC, and why this is PC1 and why this is PC4, for example, then this will become pretty obvious to you, because PC1 is the first dimension that captures the most variability of your original data set. And so because PC1 captures most of the original pattern, or at least more than the other PCs, then using values in PC1 is going to give you a much stronger signal. And then as you increase into, and if you increase or go into the other PCs, you see that they capture less and less variability in the data. And you get to a point that these PCs are really not contributing any knowledge or any separation into this original list of genes. So that comes, this goes to the question that I proposed before. How many PCs do we select? How many are necessary to maintain the structure of the data, but that is also not too many, so that they're easy to carry around? From this analysis, you can see that just eyeballing it, somewhere between PC5, maybe up, or maybe PC6 even, where we can still tell there is some separation. There is still some information about these genes. So maybe 6 dimensions will be necessary, will be good to keep. but this is, our visual estimate. And there are better ways to come up with a more meaningful number. So in order to do that, we're going to try this first method, which is called jackstraw. And we can run this line of code around line 186. I believe this step may take a little while. And while this part is loading, I already have the plot ready for jackstraw. And I just wanted to very briefly explain what we're doing with this method. The idea behind the jackstraw is it will generate a sort of background PCs, meaning it will generate this set of dimensions, based on multiple sets of bootstrapping, subsampling, and it will generate this population of PCs that are random, that are not really meaningful. So PCs that are more similar to this PC14, PC15, PCs that don't really contain much information within them. So jackstraw is generating all these random PCs and is going to use them as background, so that when we compare PC1, for example, to this background, we can quantify how much more meaningful PC1 is compared to random expectation. And this is what this plot is showing you pretty much. The way you understand this plot is that this diagonal line, it represents a completely useless PC, a PC that contains no information about the variants in the gene, of the different genes that you are testing. And so the way you evaluate the different PCs is that the more similar the PC is to this line, the less meaningful they are. And by generating this plot, you'll see that starting from PC1, which is here shown in this pink color, PC1 is as far as possible from this middle line, right? It's almost a completely vertical line. And then as you plot PC2, PC3, 4, 5, up to PC6, you see that these lines start as mostly vertical. They will start trailing off later on. But then there is this big jump from PC6 to PC7. And that is the whole point of this plot. We want to be able to identify where this jump happens. Because what this jump is saying is that once you include the first 6 PCs, you're already capture a great percentage of the original data. And then going to PC7, actually, you're not adding much, as much as the previous PCs before. So in statistics, this is called like an elbow point, which the definition is just like finding the cutoff at which the data looks sort of split, looks like it follows different patterns. So in this jackstraw plot, you can see that our elbow point is clearly in between PC6 and PC7. So based on this analysis, we'll conclude that we'll need 6 dimensions. And hopefully you're done running this step. Then you can also generate a plot. And meanwhile, I'll skip to our second method, which is an elbow plot, which is another approach to select how many PCs should we include in our analysis. And this elbow plot is more literal in the sense that we're really looking for an elbow in the distribution. An elbow that has the same definition as before, meaning the point where the cutoff, the threshold at which the points before look very different from the points after. So in this elbow plot, we're plotting standard deviation, and here we're showing the different PCs. You can see that also somewhere between 5, maybe 6, the distribution starts looking different. Here we have a very steady increase, and then afterwards it's trailing off very slowly. So on top of these plots, there are also ways to figure out where is the point that is changing the most. But it's also valid from just a visual analysis to come up with the number of dimensions that you may require. And in this point, in this example, it looked like 6 is probably the best number of dimensions. Is there any questions so far? Okay, see, it's 4:06. Now that we've covered PCA, now we're finally going into the clustering part. So same as before. I'm rushing a little bit through these slides because also I want to be able to finish this entire workshop, this entire tutorial, the art today. But I also don't want to skip very important concepts. So hopefully we'll finish it today, but if not, then it's fine. We'll just continue part of this tutorial tomorrow. So let's go back to our slides into our data analysis pipeline. And we've talked about dimensionality reduction and PCA. And in order to cluster cells, now we're not using expression values anymore, or CPM or scale or any of that. We're just using values in the PC space. Our next question is, we can see that some cells, some of these cells are already very close to each other, right, in this PC plot. But how do we quantify that? How do we find cell clusters? And the way we are going to do that is by first looking at this metric that we'll calculate, which is a cell-to-cell distance. And to calculate distance between cells, distance as in how similar of a transcriptome, how similar gene expression profile, how similar the gene expression profile is between two cells, there's been a bunch of methods being developed and multiple algorithms. And I think the main approach, the main idea circles around this algorithm, which is called k-nearest neighbors. So the question is, we want to find clusters of cells. How do we define that, oh, these cells, they don't just look like they're close together, but they're actually very close together in the PC space. They actually have very similar gene expression profile. So the algorithm being used for that is k-nearest neighbors. The k-nearest neighbors, it will calculate the distance between cells. It will calculate the Euclidean distance between cells in the PC space. Hopefully all of you are familiar with what Euclidean distance is. It's just the difference in coordinates across all the dimensions. And then you sum them together and get the nth root, n being the number of dimensions. And you calculate the distance between cells to cells in this PC space. And the reason why it's called k nearest neighbors is that this parameter k is user defined. So you can say, for example, I want to find the 6 closest neighbors for every cell. So the way you go about it is that here you have your cells in the PC space, meaning with PC coordinates. So you'll draw edges. So these lines connecting different cells. So for every cell, you'll connect it to the closest 6 or the closest k cells that you were defined. And like I mentioned, closest as defined by their Euclidean distance. And then the next step is that all these edges that you just generated, you don't want all of them to weigh the same. You want to make some edges more important than others. And how do you do that? You assign a weight to every edge based on the Jakarta index. The Jakarta index, the definition is the intersection between two sets divided by the union. Or like the magnitude of the intersection divided by the magnitude of the union. What this means is that if you look at an edge between, say, this pink cluster, there are 2 cells connected to each other. Cell 1, let's say it's over here. Let me change to a laser. So cell 1 is over here and cell 2 is over here. So there is an edge. They are closest neighbors, but then you can also look at their other neighbors. And you see that between cell 1 and cell 2, they actually share many neighbors, right? So the Jakarta index will be high. If between cell 1, say 1 from the blue cluster, and cell 2, 1 in the pink cluster, they're still kind of close together, right? But the difference is that they don't share many neighbors, maybe 1. So there is very little overlap. So the Jakarta index will be low. And once you've defined, this graph and the weight of the edges, you use a modularity algorithm, one of them being the Louvain algorithm. And what the Louvain algorithm is it finds what's the optimal modularity. This means that based on this graph and based on the weight of the different edges, this algorithm will find the clusters for you that maximizes a score that guarantees that will be the clusters where the cells are more interconnected to each other using this information. And so once we have defined these cell-to-cell distances, we can move into our unsupervised clustering. We'll execute the code for these 2 steps together later. And for this unsupervised clustering, we're going to use this method called UMAP. And what UMAP means is a uniform manifold approximation and projection. And I included the full name because I believe it's very descriptive and it's already telling you something about what UMAP is. So just as a brief explanation, UMAP is same as PCA, is also a method to reduce dimensions. You start with some high dimension space and then you end up with a lower dimensional space. One of the differences is that UMAP is mostly used for visualization, meaning that what you really want to get out of this UMAP is to represent your data into 2 dimensions so you can easily view the different clusters. And another difference between UMAP and PCA is that UMAP is a little more sophisticated. If you remember, PCA is trying to find the axis by which the variance of your data points is maximized. And UMAP is doing that as well, but with an additional step that is trying to learn the topology of your data set. The topology, meaning the shape, the structure of your data points, is trying to learn it so that when it reduces the dimensions, there is no loss in connectivity, meaning points that were close together in high dimensional space, they remain close together in lower dimensional space. And I found this example, which I think very clearly outlines the advantage of UMAP, where you have points that they make this S shape. And when you, and this is the distribution or the manifold or the structure of your original data point. And when you perform a PCA analysis, it's gonna find this axis, the blue line, as being the axis of maximal variance. But this line doesn't really do justice to the actual shape of your data, right? So UMAP will actually try to capture this pattern as close as possible. And it's also going to try to capture how cells are connected or are close to each other. So like I mentioned, so if they were close together in high dimensional space, they remain close together in low dimensional space. So I think at this point, we can switch back to the R code and let's see exactly how our cells look in UMAP representation. So we'll start by getting our distances. Here, this step is performing the KNN, which means the nearest neighbors. And here is performing the modularity algorithm to find the clusters. And if you see in the output, here is showing you the different barcodes, so the different cells, and this label, which indicates the cluster they belong to. So this cell is in cluster 2, cell in cluster 3, and so on. And once we have this clustering information, we can use it to feed our UMAP and to perform this dimensionality reduction and this 2D plot. Right, so we obtained the UMAP object. We included it into our pbmc object. And then we can proceed to plot it. Let's make it actually a little bit bigger. And here we have the representation of our original set of cells into a nice 2-dimensional space. And you can see that they are colored based on different clusters. These clusters we determined beforehand with the KNN algorithm and the modularity algorithm. And then the UMAP projected the high dimensional space, because remember, we're using multiple PCs. I believe we found that 6 was the base number, but I think in this tutorial, they actually used 10, 10. So from this 10-dimensional space, we were able to reduce it even further into a 2-dimensional space. And now we can finally see that how many different cell populations we have, in this case, 8. And so for our data set, we have 8 different types of cells. And for the interest of time, I think we can stop over here. And there is still one more section that we're missing, which is about the marker genes. But I think it will require that we spend a little bit of time. So we'll stop over here. We at least get to the one of the final steps in our data processing, where we can actually identify our cell population. Tomorrow we'll see exactly what type of cells they are. And before you go, we still have 9 minutes. So let's do a quick show of hands if you would like to do the Kahoot in these 9 minutes, or if you just want to call it off for today. So show of hands if you want to do the Kahoot. Okay, nobody. Cool. Yeah, so yeah, we'll call it off. We'll see you tomorrow, and we'll finish the last step of this, our tutorial.